So open lab stage 1 in Colab. So now we are on lab stage 1 single participant page, and we want open lab stage 1 in Colab. Here we are to build our research workflow that will contain all 4 steps: data acquisition, data analysis, data visualization, and data documentation. Let us connect our environment. Okay, so this is um, a good warning message to talk about. Whenever you close your Colab uh, notebooks, if the session in those if the sessions of those notebooks were not disconnected, then they would remain active for quite some time, or for just for some time. And so you cannot have too many active sessions at the same time. So the Colab environment will warn you that you first need to deactivate some of the already existing sessions before starting a new one. This is easy to do. You just click Manage Sessions, and you can see that here we have our sessions from the previous pre-workshop notebooks. Since we're done with those, we can just terminate them. No active sessions. And now it should allow us to connect. So in this notebook, we are going to work with a real EEG dataset. Previously, we only looked at generated data. Here, it's a real dataset that contains EEG signals recorded from participants playing emotionally different computer games: boring, calm, horror, and funny. And in this notebook, we will download and analyze data only from one participant, participant SO1. Whereas in the second notebook tomorrow, we will look at analyzing the whole data set altogether. And here we are using EEG as an example, just to simplify data processing. And Ajay in his room will cover fMRI data pipeline and fMRI data processing. So if you're consistently working with fMRI processing, you can switch to Ajay's room. And if you're more interested in EEG, you can look at this example. So let's take a look at the data set itself. The data set is stored on Kaggle. It's an emotion recognition using EEG and computer games data set. So it includes games-based EEG signals from 28 subjects. And then you have the specification of data recording strategies. So each game was recorded for 5 minutes. And so the 20-minute long EEG recordings are available for every subject. And then we have some information on how data are stored. So if we go here, we can see that the total size of the data set is 2.4 gigabytes. And if we look at the exact folder structure, you can see that the data are stored by participant, O1, O2, O3, and so on up until 28. And each folder for each participant contains pre-processed EEG data, raw EEG data, and the ratings. And again, the description includes details on what the ratings are, what the exact preprocessing steps were, and so on. The preprocessed data folder contains the data both in the CSV format and in the MAT format, so we can load either of them and work with either of them. And here you can see that all the games are stored in separate files. So game 1, game 2, game 3, and game 4. So this is a pretty neatly organized data set, in a sense that the data structure is very approachable and easy to process. So we will leverage that. All right. So here in this notebook, we are provided with prompts to um, to you to give to that we can give to the AI to complete these challenges. But in the following notebooks, writing prompts will be one of the exercises. So here, first we want to execute the model for or implement the model for data acquisition. So we want to download and analyze data from a single participant. Let's see what the prompt says. So we want a function called getData that takes data config below and reads each file with pandas, which is a Python library, using URLs, the provided URLs, and then it adds a game identifier to each file. And then it concatenates all the game files into one table and returns it. So let's take a look at what's going on here. We have our data config, which specifies the game types, and then it specifies the URLs that store the files for each game. So if we copy paste, for example, URL number 1, you can see that it's just a raw CSV file with data. So what we want to do is we want to prompt AI to get these data for every game type for this single participant, concatenate all 4 games into one table, and obtain this table. And again, we want to stay modular. So we want to have a function that does that, as opposed to that script-based paradigm that we used in the latest pre-workshop notebooks. So let us just copy paste this prompt and see if the AI can do this. And importantly, here, notice that it says copy and paste the code block from the cell below and the following text block. So we want to use both the code and the text, and we want to give both to the AI because the prompt here, it uses parts of the pre-written code. So if we just give the text, the AI won't know what to do with this data config and how it's structured and so on. So we give it the text, we give it the code, and then the AI generates the getData function, and it shows us how to use it. This is very similar to how we did it in Colab number 2, I think, where we dealt with similar functions, and then we just called them. As you can see, since the function is written this way, if we are to define another function for reading data, we would be easily it would be very easy for us to swap the data reading function later on to read maybe from other subjects or even from other data sets. Okay, so get data. Let us run this first code cell and then our get data function. And let's see if it works. As you can see here, the provided code matches what the AI suggested in the prompt window on how to use the get data function. Perfect. So it read the 4 data frames, the 4 tables from those URLs, and concatenated them into one long table. As you can see, this table contains columns corresponding to the EEG electrodes. Then it has some columns with NaN values called unnamed 14. It is probably some processing artifact. Well, I know for sure that the unnamed 14 appears in the original data, and they probably didn't process it correctly or something. In any case, we're going to drop this column later on. We're not interested in that because, yes, it can cause errors moving forward. And then it added a game identifier. Yeah, Evie, good suggestion. It might be the ground electrode. We might need to look at the raw data that they provide to fully figure out what it is. Okay, so in terms of the data analysis step, we are to assume that the columns of the data frame are recordings from individual electrodes, because that's what they are. And we have the identifier saved in the game column. So we are to write a function, run ANOVA, that takes each electrode in the data and runs a simple ANOVA between the games. And then we are to print out the composite significance results table. Now, so for real pipelines, this of course will not be sufficient. This is not a really good move to make because we're not really doing any post-processing. We're not really first looking at the data to understand if what we are to do even makes sense. We're just running ANOVA on the raw data, and we'll probably get bad results. Bad in the sense, we probably won't get any significant results, which is fine because this is an educational pipeline to just demonstrate that to just demonstrate the caveats of the AI generated analysis. So in the real world, we would of course not do that this way. We would probably first look at the data, find electrodes of interest, maybe re-reference to some reference of interest, maybe band pass filter some of these signals to analyze frequency bands of interest, uh, search for artifacts, and so on. Here we will skip this pre-processing steps for simplicity and try to use this ANOVA on the raw data. And again, note how we want to make it modular. So we want a separate function, run ANOVA, that takes the data table and produces the ANOVA results. Let's do this. Okay, let's copy paste and see what happens. Yes, so if we look at this function, it detected our electrode names as specified, AF3 through T8. And then for each electrode, it goes ahead, it takes the electrode's data for every game, and then it runs an ANOVA across those four groups, and then it tests at the 5% significance level. Note how we did not specify the significance level. It just came up with a 5% significance level, but well, this comes back to the exactness caveat that we can, of course, specify this procedure to make it more exact. And well, as expected, we don't find significant differences between groups. Um, Typical for data like this in a sense that we just essentially gave some data chunk to the ANOVA without first looking at it, pre-processing it, and so on. So it's probably really noisy. That might explain the lack of significant differences. But uh, the AI did its job and produced a significance table, which is nice. In terms of visualizing the results, let us see. We want to use a function plot results that takes the data frame and makes a Seaborn. Seaborn is a Python package. We could exclude this uh, package name and just say that we want a histogram showing averages and standard error of means. It should take the ANOVA result table from the previous step and indicate significance with stars and not null significance with an S. Let us do that. Now this one might be tricky because plotting libraries are its own beast, and very often they change very rapidly, and so the codes that the AI generate might not work. And if they don't work, the only solution really is to continuously reprompt it until it works. So let's see if this chunk that it generated here works for us or not. Okay, all right, it worked, which is interesting because when I was testing this notebook, I think a couple of days ago, it didn't work from the first attempt. I had to basically go ahead and regenerate this prompt multiple times for it to just finish running. But I guess the prompt is specified correctly now or sufficiently well now so that it generates the plot results. Well, we can see our results here. So it plots the average values of the signals and the standard error of means for each signal across all of our electrodes. And well, we can see here that the standard error of means, errors of mean are huge. So that explains um, our lack of significance. But at this point, we can go back and look at the data, figure out why exactly the standard errors are um, so huge, and we can do something about it. And finally, the fourth module is to generate um, a report for our analysis. So we are to assume that the ANOVA table from above is saved in the ANOVA result variable, and we are to write a documentation logging function. It should be called document research that creates a data frame with the following columns: current date, number of significant electrodes, and that's it. And then it should return this table. This is an example of what this table should look like. So it's just our current date when we run this notebook and the number of significant electrodes identified. Let us see. Okay. Once again, I emphasize that we are doing it in a modular fashion so that we have our document research function, not just, a black box containing all the four stages. Okay, something went wrong. Oh, okay. The prompt is misspecified. This is interesting because the function is called, there's a typo in the function name. Um, you can see it here. The research is spelled incorrectly, and it's actually spelled incorrectly in the prompt itself. That's why um, the AI complied and called it this way. Let's correct it. This is an easy error. And we can identify this error because, well, it throws the name error. It says document research is not defined when we try to call the document research function. And, well, we can see that this is because the function is named differently. So let us just rename it to document research. Okay, great. So current date is saved. Number of significant electrodes, 0 is saved. Let me correct it in the prompt itself too. All right, so we have our 4 steps, and now we have the embedded playground for you to use actually. So it has several tasks here. Well, task 1 and task 2, 2 tasks for you to play with. We're not going to go over them right now. This is for you to play with after the workshop to get more familiar with prompting and using Colab as a computational environment. But one thing to address here is that since we wrote our pipeline in a modular fashion, now we have a really clean way to execute and swap different modules of this pipeline. You can see here that our whole pipeline is essentially these 4 functions. So we get the data, we run the ANOVA, we plot the results, and we document our research. Um, and since everything else is defined above, I mean, all these modules are defined above, now if we want to change something, we just define or redefine some of these functions without touching anything else, and then it will just work down the line. We don't have to deal with potential errors. So the tasks to complete here are um, to hypothesize which 2 game types might produce different emotional responses and rerun the analysis for these 2 game types, because currently we are bundling together multiple game types, and it might be easier to separate differences between, to only focus on 2 groups. And this goes into task 2, where we can find only the games of interest and then look at the data for these 2 games, again, potentially with the help of AI, and add some basic pre-processing and see if it might improve the ANOVA results and maybe produce some significance. One thing I would recommend here is to go back to that idea that AI is really good at finding patterns. So, and to leverage the fact that these are open data. So it's safe to give this data to the AI to analyze too. So you can describe your situation like, I am working with this data set, I am doing this and this, and I'm not finding significant results. What am I doing wrong? Or what features from the data I can extract um, to enhance my analysis, or maybe should I use some different pre-processing strategies, and so on. The AI will probably identify the caveats that impact the noise levels, or maybe it will suggest some interpretable features, and we can run ANOVAs on these features and see if they make more sense. So that leads us to the end of Colab number 1. Um, Let me know if you have any questions about this. And if not, please feel free to go back to the main room and we will have some closing remarks there.